For the Kazakh people, the vast expanses of the steppe have been more than just a place of residence for centuries. The land provided food, clothing, and medicine, in the literal sense. The herbs growing in Kazakhstan possess unique healing properties. Today, scientists in the country's central region produce a wide range of phytopharmaceuticals that can easily help boost your immunity during the autumn days. So, how does an ordinary bundle of plants turn into pills and injections? Meet Artemisia glabella, smooth wormwood. Although this plant is likely familiar to everyone and needs no introduction, its medicinal benefits go without saying. But did you know that it is from a variety of this wormwood that the domestic immunomodulatory drug Arglabin is produced, a drug with no global equivalence? It is made from local raw materials. Only smooth wormwood, which grows exclusively in central Kazakhstan, can serve as the basis for the medicine. First, the herbs must be harvested, and this is done the old-fashioned way, by hand. This is where the extraction of medicinal compounds from the plant material takes place. Put simply, all the necessary components are being extracted. First, the raw material is poured into a sleeve, essentially a metal vessel. Then, the assembly is placed directly into the extractor itself. This is a device used to separate liquid or solid substances using selective solvents, known as extractants. Using supercritical high-pressure pumps, liquefied carbon dioxide is fed into the system. The minimum temperature and pressure must be 31 degrees Celsius and 71 atmospheres to transition the dioxide into a supercritical state. Once these parameters are reached via the pumps, the process itself begins. Liquefied gases in a supercritical state are used here. This state possesses enhanced component extraction properties and serves as an excellent solvent for many substances. Additionally, the carbon dioxide we use for this is an environmentally friendly substance that is safe for both humans and the environment. Furthermore, it is very accessible. The result is a slurry, a thick, concentrated extract. Just imagine, the concentration of beneficial properties here is 30 to 100 times higher than in the original raw material. But that is only half the battle. All components must still be separated to isolate the most important one, Arglabin. This is done in the next department, the preparative chromatography unit, using a specialized setup familiar to any chemist. It is called a centrifugal partition chromatograph. It consists of a single rotor, which is a type of column. This, in turn, consists of multiple stacked discs containing small cells. It is within these cells that the chromatographic separation actually occurs. The stationary phase is pumped into these cells. It is held in place by centrifugal force. That is, when the unit rotates, the centrifugal force is created, and the liquid phase inside the unit becomes stationary. Next, we inject the carbon dioxide dissolved extract and introduce the mobile phase. Based on the solubility of these carbon dioxide extract components, they are distributed between the stationary and mobile phases. The operating principle is the partition of substances between two liquid phases. In other words, like dissolves like. Since the main impurities in the carbon dioxide extract of smooth wormwood are waxes and paraffins, the system is designed so that these interfering ballast substances remain in the rotor, while the target component, Arglabin, exits with the mobile phase. Next, the resulting solution is sent to the recrystallization unit. It looks like this. The oil-free Arglabin is simply placed in a cold storage chamber. Then we filter it and process it with acetonitrile. After acetonitrile comes alcohol to remove any residual oils that remain within the Arglabin composition. Then comes the final stage, hot hexane. The output is pure native Arglabin, white crystals. The next stage in creating the future drug is the solid dosage form unit. This is where the encapsulation process occurs. A mass consisting of excipients and the Arglabin substance is loaded into the mixer. Everything is mixed for 45 to 60 minutes until it reaches a homogeneous state. The mixture is then poured into a collection vessel and transferred to the wet granulation unit. At a speed of 3000 rpm, using an anchor-type agitator, the mass is moistened and broken down into small granules. The entire process is automated. Next, the finished mass is loaded into the hopper of the capsule machine. Meanwhile, empty capsules are fed into the tooling. Using a vacuum, the cap is separated from the body of the future pill. Then, the capsules are filled with the substance, and the cap closing process occurs again under vacuum. The next stage is packaging, which takes place on a blistering machine. Using compressed air, the base is applied to the foil backing. Under a temperature of 115 degrees Celsius, the cells, pockets, are formed where the drug is stored. The quality control process is quite interesting. A medium mimicking the human stomach environment is created in special containers. Within 1 hour, the Arglabin capsule must dissolve. Most importantly, Arglabin is also intended for the immunorehabilitation of oncology patients undergoing chemotherapy or radiation therapy. Thus, a medicine made in Kazakhstan and derived from ordinary wormwood is saving lives.